The endocannabinoid system is at least 600 million years old and is present in every animal species except insects. The discovery of the endocannabinoid system began with the search for the active ingredient in cannabis. CBN was the first phytocannabinoid to be isolated at the end of the 19th century, and its structure was identified in the early 1930s by R. S. Kahn and was chemically synthesized by R. Adams in the US and Lord Todd in the UK in 1940. The second phytocannabinoid, CBD, was first obtained from cannabis that same year by Adams and colleagues, and THC was first extracted from cannabis in 1942 by Wallner, Mitchett, Levine, and Lowe. In 1963, the structure of CBD, and in 1964, the structure of THC was described by Raphael Mechoulam. In 1988, cannabinoid receptors were discovered. After the discovery of these receptors, the question remained whether mammalian tissues also produced a molecule that would interact with these recently discovered receptors. This led to the discovery of the endocannabinoid system. The endocannabinoid system is comprised of cannabinoid receptors, their endogenous lignans known as endocannabinoids, and the enzymes responsible for both their synthesis and breakdown. The endocannabinoid system plays a crucial role in regulating multiple physiological processes, and endocannabinoid receptors are found throughout the human body, including the nervous system, internal organs, connective tissue, glands, and immune cells. The receptors identified in 1988 were later termed CB1 and CB2. These receptors are found in the cell membrane. These receptors are called G-protein coupled receptors. CB1 is the most abundant G-protein coupled receptor and is found in major brain regions that play a role in learning and memory, motor function, anxiety, stress, and smell. CB1 receptors are also found in the lungs, vascular system, muscles, gastrointestinal tract, reproductive organs, fat cells, and liver cells. CB2 receptors are mainly located on immune cells. The number of CB2 receptors increases up to 100-fold following tissue injury or during inflammation. But there is an overlap in the location of CB1 and CB2 receptors. In 1992, the first molecule produced by the body that was shown to interact with the CB1 receptor was identified. It was named anandamide from the Sanskrit word ananda meaning bliss. The second molecule identified was 2-AG. 2-AG is the most abundant endocannabinoid found in the brain. It plays a significant role in the development of the central nervous system and influences brain plasticity. Other receptors involved in the endocannabinoid system include receptors such as GPR18, GPR55, and GPR119. Several other molecules interact with the endocannabinoid system, such as P, and the endocannabinoids are synthesized on demand, as compared to other neurotransmitters which are stored at the end of neurons, and endocannabinoids work in a retrograde manner. Normally at a synapse, a neurotransmitter is released from the presynaptic part of a neuron and diffuses to the postsynaptic part of a neuron, where it binds to and activates a receptor. In retrograde signaling, a messenger is released from the postsynaptic part of a neuron and travels backwards across the synaptic cleft, where it activates receptors on the presynaptic part of a neuron. Studies indicate that when CB1 receptors are activated, they can inhibit the release of several different types of neurotransmitters, so the endocannabinoid system can act as both an excitatory or inhibitory system.